Research is not for poor people nowadays, it's for the rich people business, you know.
Good morning everyone. Today I am going to show you an experiment where I will be throwing sawdust of wood samples on malt extract agar plates. The purpose of this experiment is to see what kind of microbes or microorganisms we can get on malt extract agar plates, because we are working on an experiment where we need to see those microbes in contact with the pathogen. And so let's start our experiment. I will guide you step by step.
Okay, so this is my table and you can see here some plates. Actually they are not malt extract agar plates, but they are nutrient agar plates. But what I will be doing is that I will fill, these are the empty Petri dishes, so I will be filling these dishes with the malt extract agar. Okay, the first step to start anything in the lab is to wear the gloves. So here we have different sizes of gloves, as you can see. For example, here you can see M, which means medium, and then L for large, and then there is some other sort of gloves. So you can have different kind of gloves. So this is the first step. I will wear the gloves and then I will start preparing the malt extract agar. Now that I have my gloves on my hand, and this is the weighing machine where I will be weighing different compounds, for example malt and agar, to put into these bottles. First we will check this weighing machine. As you can see here, this bubble is not inside the circle, which means that it is not balanced. So first we will try to make it in balance. Alright, now you can see that this water bubble, let me emphasize this, yes, so this water bubble is inside this black circle, which means that now our machine is balanced, and now we can turn it on with this button. Alright, so now we can start weighing our compounds. Let me take you to take the malt extract, because we need to keep malt extract in the fridge, otherwise it can degrade easily. So this is our lab fridge, and here we have, you can see malt extract. I'm taking this with me. Oh wait, I need also something on which I can weigh. First I will close this door. Okay, I have cut this piece of aluminium foil because I will be using it to put this malt extract on it. So let's go to the other room in our lab where there was a weighing machine or the scale. So here is the weighing scale. What I will do, I will just take this out because of the surface that we can increase, and then I will be putting this foil on this, and now you can see that the weight has been increased because of this foil. So what we have to do, we have to press this button. Here it is Tare. So now you can see that when we press this, it reduces the weight to 0 grams. So actually we are going to prepare 2% malt extract, and now I have measured 20 grams of malt extract, and then I will be pouring this into these bottles. Like this one is empty, so I'm going to put it here. Okay, now that I have put the malt extract into these bottles, I have to take 15 grams of agar to mix into that bottle. So I can take this. This is the agar, as you can see. And I will measure 15 grams on this sheet. Okay, now as you can see, I have measured 15 grams of agar. It's a little bit above, but it's okay.
and I will be taking this and pouring into these bottles. As you can see here, this yellow part, this one is actually malt extract, and this whitish part is agar that I already poured into one of the bottle. Now I will be pouring that portion into this bottle. Okay, you know what, now it is already there, my malt extract along with agar. The next thing I need to put into that bottle is magnetic bar, because we need to stir along with water so that it can be mixed. So we'll take these bars, magnetic bars. The purpose of including these magnetic bars is that I will just show you. Wait a moment. I put one bar here, and another bar I'm going to put into this. Before I explain, I turn off this. So purpose of this magnetic bar is that when I will put the water inside the bottle and I will place it here on this machine, which rotates this magnetic bar, and it helps in dissolving malt extract and agar into the water. Let's see if it can work even in the dry condition. I just turn on the machine. Okay, so now it is at 0. If I will increase, probably you will see the movement into this. Yes, so you can see that the magnetic bar is moving. Okay, now I have filled these bottles with the water, but this water is not tap water, is a little bit distilled water. So now let's start the machine, and you will see that the water will show some movement. So you can see that it is moving the magnetic bar inside this bottle, so that the dissolution of agar and malt extract could be made uniform. Alright, so now after dissolving these the water with the agar and malt extract, you can see on that machine that I showed you in earlier step. Now I'm going to place them inside autoclave. And remember that when you place these bottles, you need to make sure that the lids are a little bit loosen up. So let me open them a little bit. So they should be loose, not so loose that they will be taken out with the pressure of the water, but just a little bit okay, now it's good. We will place it here, and we will do the same thing with this bottle. Alright, so now we will close the lid. This is an old autoclave in our lab, but still is working, so right, we don't need new one. We don't need to waste money. Research is not easy. Research is not for poor people nowadays, it's for the rich people business, you know. Okay, so another thing, important thing is that now this is closed, this valve. I don't know if you can see, there is the hole. So now we have to turn it down and start the machine, and you can see this meter. Now it is at 20. When the temperature reaches 100 degrees, then I will close the valve so that the pressure inside the chamber will elevate, and until the needle will go to 120 or 22 degrees, I will let it for at least 15 to 20 minutes, and then I will turn off this button and wait for some time, probably around 15 minutes, and then after 15 minutes I will slowly
again open this valve so that the hot air from this area will release from the chamber. Alright, so now the bottles have been sterilized in the autoclave, and I have put them in this hood, and these are the Petri dishes. Now I will cut the bags of these dishes, and then I will pour into these Petri dishes this Malt Extract Agar media. So, and I will show you later when I will be I filled all these dishes. So I will start and I will show you the end result. Alright, so now I have poured that malt extract medium into these Petri dishes. So you can see that these dishes are filled with the malt extract, and now later I am going to put sawwood or wood powder onto these plates, and I will show you the end result, how it looks like. Alright, so now I have already sprinkled sawdust of wood on my plate, as you can see here. So after 2 or 3 days I will come and check it again if something grows out of this sawdust, and let's continue in the next video what we will be having the results. Till then, bye bye.